Thanks very much to the organizers to invite me to Berkeley. I feel like it's the home of Twitter. I think about half the people I follow seem to be from Berkeley. And the Uber app has lots more features in San Francisco as well. I'm going to talk about a class of modeling strategy which we're using a lot. So it's a kind of something that I think people should consider adding to their toolbox for analyzing data. We found it to be very useful, and I'll give some examples of different ways we've been using it. I'm going to talk about Gaussian processes, which are a way of doing non-parametric regression. I'll give a quick intro, and then I'll discuss a bunch of different ways that we've been using these in different kind of applications to genomic data. So a Gaussian process, for those with a physics background, they'd be familiar with these as models of stochastic processes. But in statistics and machine learning, they become synonymous with various types of regression and classification tasks. And so we think of them as very flexible functions which you can fit to data. And the key object in these Gaussian processes is the covariance function. And the covariance function determines the properties of the samples from this distribution. So it's a distribution over functions, analogous to a Gaussian distribution over numbers. So you're drawing these functions, and they can be static or dynamic, smooth or rough and stochastic. They can be stationary, non-stationary. They can have structure like periodicity, or they can be very kind of chaotic. And so the covariance function has a form and parameters which kind of tune the behavior of the functions that you draw. So this is a random distribution over functions. So let me give you an example. So this isn't a Gaussian process. This is just a regular Gaussian distribution. Okay? But it has a very specific covariance matrix. And I think I missed Monday, but I think you heard all about covariance matrices on Monday. So we choose a covariance matrix which has this high kind of diagonal here. And that means that things are close together in these 25-dimensional vectors. So I'm going to draw 25-dimensional vectors from a Gaussian distribution with this covariance. Indices which are close together will be more highly correlated. And when you do that, the 25-dimensional vector, if you plot it along the index like this, it starts looking a bit like a function. So, if you join the dots, it looks like some kind of smooth curve. So a Gaussian process is just where you take that limit formally to infinity. So you no longer have a Gaussian distribution over 25 points. You have a distribution over these infinite dimensional vectors or functions. And the way I've generated these curves in the computer is actually by generating Gaussian vectors which just have so many points that you can't make them out anymore. So these are just 1,000 points instead of 25. These particular curves were sampled from this covariance function. And so this particular covariance function generates smooth, infinitely differentiable analytic functions which are marginally Gaussian. So it means that if you look at a particular index, the distribution of the values you get here for this function follow a Gaussian distribution. And any finite set of indices is going to generate something from a multivariate Gaussian distribution. And that's the definition of a Gaussian process. Any kind of finite set of indices I take has a jointly multivariate Gaussian distribution. And that makes it very convenient to compute over these objects, because we can do all sorts of nice computations with Gaussian distributions. So Bayesian Regression 101 with a Gaussian process. Before I see any data, so I'm being Bayesian, I already have some pretty strong beliefs over the kind of thing I'm going to see. So So I think I'm going to see some population gene expression data. So it's going to be smooth because it's averaged over millions of cells, okay? And so I'm going to hallucinate some kind of gene expression profiles. And these are draws from my prior distribution. Okay, so this is before I've seen any data. I believe I'm going to see these kind of wiggly functions. When I see one clean data point with very little observation noise, then I'm constrained by the data and my belief. So now I believe that I've got these smooth functions, but they're going to go close to this data. And the more data I see, the more the data constrains me. And so my posterior distribution over the functions that go through this data becomes closer to the data, and the posterior variants, or the credible regions, become tighter. And then if I have a lot of data, I see that this was actually generated from a sine wave. And the non-parametric nature of this kind of regression is quite powerful because it's very flexible in the types of functions that I can fit here. But the prior is also quite strong because my prior was that the curves would be smooth, these kind of analytically infinitely differentiable functions. And so there is a strong prior, but it has this non-parametric flavor that I can model quite arbitrary shapes as long as they have the right kind of smoothness characteristics. Okay, so my first kind of application is to modeling batch effects in kind of time course data, and then how to do clustering while taking into account those kind of batch effects. So this is data from this nice paper about gene expression in different Drosophila species, but we're just going to look at one of the species. And you can see that these are these different batches where they did these time course experiments. But I think at the point where they got to the fifth one, they thought, oh, we better do a bit longer because the biology is getting kind of interesting at the point where we stopped doing it. So they did a few extra time points, but then maybe their budget ran out. So then they thought, well, we better not do any more of these early ones because, you know, we haven't got budget. So we're just going to do the ones at the end here. And then they give the data over to the bioinformatics team. And they're like, what? You know, what are you doing? And then we come in and say, oh, this is a great example for Gaussian processes. So we can kind of deal with this. So, you know, the usual thing you might do is just kind of lump all the data together. You might just concatenate all the time series, or you might average. If you lump it together, you're losing all the within-batch correlations, which in time series are incredibly valuable. You know, so you really don't want to do that with time series. You don't want to throw away all that valuable within-batch information. If you average, you're losing your information about what that batch variance was. So you're also throwing away a lot of information. So those two approaches are very suboptimal. So instead of that, what you can do is you can try and model the batch variance. And we use a kind of additive hierarchical model. So you have your kind of mean behavior of the gene across all the batches. And then each of your batches is another Gaussian function with that kind of mean behavior. So the mean of the Gaussian is added to by another random function which catches the within batch kind of behavior. So it's a kind of very structured noise model, if you like, with temporal correlation. And then you get this kind of covariance matrix here. And it allows you to basically model the variation across all these different batches. So you can see, for instance, this one here comes down much further than this one up here. And it's a temporally correlated kind of structured batch effect. So you can kind of model that. You can also extrapolate away from the data by borrowing the information from the other replicates. So even for the cases where you only have data at the end, you can make predictions about what would have happened if you'd had data earlier. And actually, there are quite nice machine learning applications of Gaussian processes where you can, for instance, with weather stations, deal with what happens when a particular station gets knocked out when you're measuring weather patterns across the ocean, and you can kind of use this borrowing of information from the other weather stations to kind of impute the data that you're missing. So that's quite a nice feature for missing data. You can go on and extend this hierarchy to do clustering. So now, instead of just having genes and replicates, I also have clusters of genes. And now what you do is you have all the different genes in a particular cluster. This is a cluster behavior, but you also have all the replicates along here which you're modeling. So you can really capture this rich kind of covariance structure of the genes within these clusters. And we kind of plugged that model into an existing probabilistic clustering algorithm for this particular, for the same data. So it's exactly the same algorithm that someone else had used, but we just plugged in our particular covariance model. And it really changed the clustering. We got fewer, which were more correlated with kind of biological annotation. But that particular algorithm wasn't too fast, so we also developed a very fast version of a Dirichlet process mixture model, a non-parametric kind of mixture model, so that you can do this genome-wide. And there's some nice code for that, which James Hensman has produced, and we have Jupyter notebooks where you can play around with that stuff, which James has also done. Oh, one thing I should say, a nice thing about using this kind of model-based clustering is that you can put structure into your cluster models. And one thing that people often want to do, for instance, is model periodic data. And so an experimental group in Manchester work on circadian, sort of tissue-specific circadian clocks, took our clustering algorithm, and then we put a periodic Gaussian process covariance into that, and did some clustering over their kind of periodic gene expression data.
Magnus?
Yeah.
Can you specify the period, or can you put a prior on it?
In this case, we learned it as a hyperparameter, but I think we had a prior to keep it within the kind of circadian range. Yeah.
When you say you learn it, you somehow try to maximize the total evidence summing overall possible.
Yeah, yeah. Actually, we have a nicer periodic covariance function now, which has just been published, which allows you to model very non-sinusoidal periodicity, like top hats, sawtooth, all that kind of stuff. So it's really nice for non-sinusoidal. A lot of the methods for detecting circadian genes, for instance, are highly biased towards sinusoidal genes. But clearly, lots of circadian genes aren't sinusoidal because you use the core clock to decide when to some specific time when you want to turn something on, like 4 o'clock in the morning. So lots of circadian genes aren't sinusoidal, and I think most methods are highly biased towards sinusoidal rhythms. So there's a nice preprint that you can get. Okay, so the next example is branching and bifurcation kind of processes, and this is quite recent work. So the first example is where you have two time course, and you want to determine the point at which they begin to diverge. So basically, you do an experiment where you have a perturbed system and an unperturbed. You take a time course, and then you want to work out where's the first point where the perturbed time course deviates from the wild-type behavior. And that's really actually very easy to do with Gaussian processes. It's very natural. And so the kind of model I'm going to use now is one where you have a Gaussian function, but at some point, another Gaussian function branches away from it, but they touch at this particular point here. So we can sample from the prior. So this is our prior. Here's another sample. Here's another sample. Okay? So these are all Gaussian functions which touch at that particular point. And you can work out the covariance for two functions crossing. Imagine you have two Gaussian samples from Gaussian processes, like this, and they cross at a very specific point, and that's the constraint that those two functions have to cross there. And then imagine you have two data sets, so you have some green ones, and they go along here, and that's a wild type. But then something happens to the other data set at the crossing point, and that data goes along here. So the crossing function covariance is this guy here. Oops. But then if I have some data which comes from the joint part first and then splits, then this is a covariance of that data that comes from that kind of splitting process. And the nice thing about this is it's exactly tractable. So I can actually work out the likelihood of this, whereas previous approaches you had to use these kind of quite difficult approximate inference techniques. This is exactly tractable, this model. And so I can just work out the likelihood for all possible splitting times, and I can just compute the posterior exactly by just doing a histogram and just normalizing. So there's no approximate inference, no EP or variational Bayes or anything like that. It's all exactly tractable Bayesian inference, which is quite unusual, actually, to be able to get away with doing something like that. It means that I can compute multimodal posteriors over this splitting point. So that's I think that's quite nice that you can do that. And it's very kind of well calibrated in simulated data from different kinds of scenarios where you have a splitting point. So you can really work out the rank order of where these splitting times are quite precisely, compared to naive approaches where you might just choose the first point where the differential expression goes past the threshold. That approach is very poorly calibrated because the way that you get false discoveries will depend a lot on where the perturbation time is. So I think this kind of Bayesian approach really is very well calibrated some way. And so we've kind of applied this, and we've got a paper which is in press, and there's a preprint on the archive where we've used it to kind of try and disentangle what happens when you disarm a kind of a pest for these kind of tomato plants. And you can kind of look at this as a new way of doing differential expression analysis in this kind of data. But what I'm quite excited about is that this branching Gaussian process, you can actually generalize it to many different branches. You can work out the marginal likelihood, so you can actually search for the optimal number of branches in this kind of data. And a nice application of this is to single cell data. Okay, so with single cell data, you often have some kind of branching dynamics, and you want to kind of learn about what the branches are, where they are, how many branches there are, and so on. And that's what we're currently applying that branching model to.
What are the axes on that plot?
Oh, this is a projection using something called a Bayesian GPLVM, which is a nonlinear PCA plot. So this is nonlinear PCA of the cells, and then this is some kind of pseudo-time along here. So instead of a time series, we now have some sort of pseudo-time, basically. It's a much harder problem because you have to infer pseudo-time, and you have to infer where the cells are on the branching pattern. So it's kind of a tough one. But that's kind of current work. A bit too new to present. And finally, these methods are also applicable to simple differential equation type models of dynamics. So in this work, we had some POL2 data and some RNA data, and we wanted to link that in order to understand the delays in the RNA relative to the POL2. And people have done this with a number of different approaches, and this is one kind of way to do it, which I think has some nice features. So you have a rate of change of the RNA is assumed to be proportional to the amount of POL2 which you have at the 3' end of the transcribed region. So you just measure pol2 after elongation is completed. And we're trying to understand how this whole process is kind of regulated. And when we did this initially, we couldn't get a really good fit for all the data. And it turned out that there was some delays in the system which we couldn't quite understand. They seemed too big. So we put in a delay term, which is some kind of delay that happens after elongation is complete, but before you see the mature RNA. So it could be to do with transport at the nucleus, or it could be something to do with release. Or we don't know exactly where that delay is in the system, but we just put it in as a parameter to infer. We model the pol2 profile as a Gaussian process. And then it turns out that the whole thing is exactly tractable, and you can work out the joint distribution of the mRNA and the pol2 as a joint two-dimensional Gaussian process. So everything's exactly tractable in terms of the likelihood. I'll just show some examples. So we're interested in this delay parameter. This is the posterior distribution for the delay. So this is a gene which is quite early active, but there's no real evidence of delay here. And then this is one of the ones which looks like it's got a delay. The pol2 comes up, then the mRNA comes up, and it can't be explained just by the half-life of the mRNA. It looks like there's something in the system which is really delayed in this particular gene. But then that one has some evidence of a delay. If you look at some genes, though, it's not trivial, because the mRNA peak can be after the pol2 peak in genes which have no evidence of a delay because they have a stable mRNA. So the half-life here is quite long. So that means that you get a delayed peak in the mRNA relative to the pol2. So that's why the differential equation is useful to interpret this data. But you can see the data is very noisy. And so this kind of probabilistic approach is quite useful for this kind of data. You can't just join the dots and with this kind of data. And so we found these really large processing delays in about 10% of genes. And we looked for correlations of different genomic features and so on, and we found that the highly delayed genes were enriched for short genes. So often it was short genes in terms of the pre-mRNA length, which were delayed. And there was also some intronic features which were kind of correlated with delay. And we suspected that perhaps some of these short genes, so we felt that with the short genes, elongation would be very quick, because elongation sort of happened in about 4 kb per minute. And so these things are basically elongating in about 2 minutes, something like this. But they often still have quite a complex structure in terms of introns, even though they're short. They're still long enough to have complexity. And we felt that maybe splicing would be something involved here. And there was some evidence previously that some genes were delayed while splicing is completing. We don't know where the splicing could happen. So we looked at the intronic reads in our data because we had total RNA-seq. And it did seem that there was evidence that the delayed genes tend to have more increased retention of late introns over time. So we came up with a kind of metric for determining intron retention over time. And it seemed like a lot of these delayed ones have evidence that the introns are retained. So we think there's some evidence that, at least for a proportion of these highly delayed genes, that's something to do with splicing. But it's only a proportion. There could be other things going on. And there's some very interesting recent imaging work showing that also transport at the nucleus can take much longer than people thought originally. So we don't know exactly what the cause of that is.
But this thing is quite complicated, right? Because you have really a population of cells, and this differential equation is sort of true for a single cell, but then when you see how much pol2 there is, you're looking really at the sort of a population average.
That's true. I mean, this is a highly synchronized scenario where you take a bunch of cells and you hit it with a stimulus. And so the short ones are the ones which are actually doing stuff quite early, usually. So we're hopefully still in a fairly close to the highly synchronized part. But you're right. I mean, everything is smudged out a bit by the fact that you have a cell population. Actually, we have a model of elongation, which I'm not talking about, using Gaussian processes, where we explicitly model the spreading among the cells in a kind of elongation model. And we needed to do that to get a robust result for the Pol2 moving along in long genes. So yeah, that was really important in that elongation model. I think because this one is a shorter one, the signal's a bit sharper. So, okay, so how important is it? Well, elongation time seems to dominate for most genes over this kind of delay that we saw. But there is a sort of subset of genes, over 10% of genes, where it seems that this delay that we see is actually more important than elongation time in terms of determining how long it takes to make the mature RNA. So it seems like it's a kind of significant effect for a proportion of genes. We can also extend this for time-varying degradation when that's under regulation. But that's quite challenging because things become non-exact in the whole Gaussian process approach. And yeah, I mean, it's a bit of a whistle-stop tour of different approaches where we've used these. But I think I'm sort of selling the methodology in this talk. So I like Gaussian processes because they're got a good mixture of flexibility and tractability. Okay, so hopefully you've seen that you can do kind of hierarchical modeling very nicely with these. You can introduce structure like periodicity. The models remain tractable under constraints like branching and bifurcation. And they're tractable under linear operations on the functions. So if you do a linear differential equation or a linear partial differential equation in a spatial system, the whole thing remains a Gaussian process, so the likelihood becomes is exactly tractable in those cases. You can make compound kernels. So if you have a periodic process, but it's got a trend, then that's really easy. So it's really easy to make compound models which have features of lots of different models. And that's actually really important to be able to do, because usually a signal isn't just purely periodic. Usually it's periodic and kind of varying in some other way. Data is often non-Gaussian, but they're really good approximate inference algorithms, and a few people have recently published count-based models using Gaussian processes. So using count data, but modeling it with Gaussian processes, the two groups have just published papers modeling, I think, RNA-seq data using that kind of approach. And importantly, the code base is kind of growing. And we work a lot with Neil Lawrence's group's package called GPy, which contains a lot of different types of covariance function which you can use and adapt. And there's lots of nice Jupyter notebooks to show you how to use that. GPFlow is a very exciting development from James Hensman and Alex Matthews, where they take TensorFlow, which was designed to do kind of deep learning gradients, because computing gradients is a big part of those applications, and apply it to GPs. So that means that you don't have to sit down and do the derivatives. And actually, the differential equation work, Antti Honkala had to do about 125 terms in one of the integrals in that paper. So it looked simple, but the covariance function was quite hard to deal with. So GPFlow could really speed things up there. And there's some R packages like the ones that we've produced. And this is a project where we've collaborated a lot with Neil Lawrence, Antti Honkala. James Hensman has done lots of the work and lots of the coding here. Jing Yang did this time perturbation model, and Alexis and Max have been working on the single cell data. And that's it. Thank you.
So I'm just curious, Cohen and self-organizing networks, if a bit of the same flavor of some of the stuff. Is there a connection there, or could you think of this as a generalization of that?
So the closest connection with the self-organizing maps is the GPLVM, which is a latent variable model where the Gaussian process is used as a mapping from a low-dimensional space into a high-dimensional space. And so it has very much that kind of flavor, I guess. Yeah.
I was curious in the branching process model. So in the one you draw, for example, there is a smooth curve and then branching where the branching is not smooth, like a main trajectory and some merging. So is it part of the model, or is your model more symmetric about the two branches?
The model for the single cell data is symmetric, but the one I showed for the time series is not symmetric. So in the time series model, the wild type is smoother than the perturbed. So the idea there is that you have some kind of smooth behavior, and then you perturb the system and something shoots out of the side. You could constrain the first derivative to be the same at that point, and that would lead to a smoother branching. But you can't constrain all the derivatives because it's an analytic function. So if you constrain all, they'll be the same forever. So you can only constrain one or two derivatives. Yeah.
I was curious, a bit out of the scope of the presentation, but are there any other stochastic processes that have nice properties, like maybe for count data? Like you said, you can do Gaussian process.
So there's a lot of different non-parametric. There are kind of lots of non-parametric approaches for discrete data, not for kind of smooth functional data. I think the Gaussian processes are the most tractable for inference. Other stochastic processes, I imagine, are rather difficult to do inference with.
So actually, I'm wondering about, to follow up on that, could you think of a model where, like, the lambda from a Poisson distribution is what's modeled with the Gaussian, but then there's some kind of...
So people use Gaussian. So people use Gaussian processes for that.
Oh, that's how.
Yeah. So people do that in modeling spiking data in neural kind of measurements, and then you model the lambda from the Poisson, and you do it as a Gaussian process. And they often, if you have a lot of data, you can use these kind of path integral techniques to actually infer. So actually, when the process isn't Gaussian, in physics world, they often expand around the dominant path using a Gaussian expansion anyway, which is like a Gaussian approximation, Gaussian process approximation to something. So it's kind of related to path integrals and all that kind of stuff.
Yeah, so I also want to be about Harman's, because in the gene expression case, the way I would sort of physically think about it is that there's some smoothly varying rate of transcription, but then the actual number of mRNAs that fire, right, is some Poisson sample.
Yeah, it's quite easy to put non-Gaussian likelihood functions into Gaussian processes. So, for instance, log-Cox models and spatial statistics models often have discrete observations, and they use Gaussian processes as the model over a rate or something like that. And so you can do inference over a discrete distribution, but then the inference has to be done with approximate inference techniques if you're doing Bayesian inference. But they're fairly tractable, so you can do it. Yeah.
Okay, well, let's thank all the speakers of this session again.